research. I'm going to talk about the conserved region and the hypervariable region. I'm going to introduce you guys to the 16S microbial data. We are going to discuss the common applications of 16S RNA sequencing, advantages of 16S RNA sequencing, limitations of 16S RNA sequencing, microbiome diversity matrices, as well as differential statistical analysis. So I'm going to be a little bit fast, but if my attention is needed, I can go back and discuss further. So what is the 16S RNA gene? The 16S RNA gene is, in simple terms, is just like a fingerprint for bacteria. Every kind of bacteria has the 16S RNA gene. And even though it's all bacteria have the 16S RNA gene, some section of the 16S RNA helps us to distinguish one bacteria from another. So this is the full length of the 16S RNA gene. It consists of about 1,600 base pairs, and it's a very highly conserved gene present in all prokaryotes, both bacteria and archaea. It contains the conserved region, which are highlighted in red, the V1 to V9 regions, and also the hypervariable regions. That's the ones highlighted in gray. So during sequencing, the V3 to V4 region is mostly mostly amplified during PCR in gut microbiome research. We may want to know why the V3 and V4 region is mostly used in gut microbiome research. This is, hello?
Yeah, we can hear you. Please, Dr. Umar, please, can you mute yourself? Because I've been meeting you and you're muting yourself. Umar Mayadon, please.
So the V3 and V4 region has enough variation to distinguish between many bacterial genera and even some species. V3 and V4 region is about 450 base pairs, which fits well within the paired end read lengths of common sequencing platforms like Illumina. Databases like GreenGenes and Silver have a lot of reference sequences for V3 and V4 region, making taxonomic assignment more accurate. So these are few reasons why the V3 and V4 region are preferred in gut microbiome research. Then I will want to tell us a little bit about what the conserved region of 16S RNA gene means. We should look at the conserved region as a particular paragraph in all books you check, and that particular paragraph is the same across all books you check. So this is just an illustration to show us how the 16S RNA is used to identify bacteria. It's just a particular paragraph, just like a particular paragraph in a book that is the same across every book you come across. That's how the 16S RNA is being used to identify bacteria. The conserved region, while the hypervariable region is it differs from one bacteria to another. Just like the particular paragraph in another book, it contains another separate coding. So this is how it is used to distinguish within bacteria between different genera, different phyla, different species, and others. So with this brief introduction, I will want to introduce us to the 16S microbial data. So this 16S microbial data is the sequencing data derived from 16S ribosomal gene of bacteria and archaea, which is commonly used to identify, classify, and quantify microbes in a given sample. So how do we obtain the 16S microbial data? This is a brief workflow of how the 16S microbial data is obtained. After DNA extraction, bacterial DNA extraction from a sample, the sample is amplified via PCR, particularly the V3 to V4 region. The PCR product is being cleaned up, and library preparation and sequencing is carried out on an Illumina platform. After that, the sequences are obtained and compared to database like Green Genes and Silver to obtain the particular bacterial taxa that the sequence is matched to. Then the downstream analysis, which is what we will be doing in this session. The alpha diversity, beta diversity, differential analysis, and visualizations. These are components of the 16S microbial data, raw reads. These raw reads are FASTQ files. They are sequences obtained from sequences. This is how they look like. They are normally in notepads. So these sequences are the ones that are paired, are uploaded to databases to obtain the taxa. If the sequence is matched to taxa, then it will be able to give us which bacterial phyla, genera, or species that the sequence is matched to. So the amplicon sequence variants, ASV, or the operational taxonomic units. So this, depending on the type of sequences that was carried out, it's either ASV or the OTU. Let me briefly differentiate the OTU and the ASV. This OTU, that is the operational taxonomic units, are sequences, that's the one I showed you earlier, that are being clustered based on similarity, usually 97% similarity. So if a sequence is 97% similar to that of a bacteria in a database, it will automatically identify that sequence as that bacteria. So just 97% accuracy. However, the ASV requires exact sequences 100% down to every single nucleotide differences. So this means that the OTUs have very low accuracy, not very low actually, but low accuracy compared to the ASVs. The ASVs are highly accurate. So if the closely related bacterial species can be erroneously placed as one OTU, while under ASV, it has to go down to the strain level, or even it's exactly that particular bacterial sequence. So it's OTUs, although here in Nigeria is just upcoming, but in advanced countries, it's becoming outdated. The ASVs and shotgun metagenomic sequences are being introduced recently. So the pipelines or the common softwares used in this OTU sequence analysis is the KIME1 or KIME2, while the ASV is the DADA2. So taxonomic tables. Taxonomic tables are when these OTUs or CSVs, OTUs or ASVs are being matched to database, to NCBI or to Green Genes database. So it gets assigned to a particular taxa, that's phylum, genera, species, family, and the rest. So these taxonomic tables show us which OTU get matched to particular taxa, like this OTU1, Firmicutes, and the genera Succinylclatium. So you can look at all the OTUs being matched to particular phyla, class, order, family, or genera, species, and so on. So this is how the taxonomic table looks like, and we will be using the taxonomic table today for our analysis. Future or count table, or sometimes called the OTU table. This count table is the count of each OTU in a sample. This is how it looks like. These are the OTUs. In case of ASVs, these ones will carry ASV number. So these ones, these ones are the replicates. You can look at group A with 1, 2, 3, 4, 5 replicates. Group B, 1, 2, 3, 4, 5 replicates. So these ones are the replicates and these are the counts. So as I said earlier, OTO1 has about, in this particular replicate, has about 662 bacterial group taxa matched to this OTO1 in this particular. So these ones are 97% similar to OTO1. So this is what OTU means. Metadata or sample data. This one gives an information about each sample. This is how it looks like. This one is your information about your sample. That's the environment. If the samples are collected from different environments, in this particular example, the samples are from different groups of animals. These ones, the group, the control, treatment 1, treatment 2, treatment 3. This is the sample number. And these ones are the management system. From 2 different management systems, you can have different environments, other things, so that you can analyze based on particular factor you want to consider. So common applications of the 16S RNA sequencing. 16S RNA sequencing is important in understanding microbial community structure. It is useful in comparing microbial diversity across treatments, time points, and environments. It is useful in linking microbes to health or diseases or environmental factors. It is important in biomarker discovery. Advantages of 16S RNA sequencing. It is cost-effective when compared to metagenome sequencing. So it's relatively affordable. It's good for unculturable microbes. Maybe some microbes are not easily cultured in the lab. So the 16S is good for identification of them. It supports large number of samples when compared to the traditional culture-based method. So the limitations of the 16S RNA sequencing, it doesn't give functional data like the shotgun sequencing. However, this nowadays with the functional prediction softwares like TaxForFun2, you can be able to predict functions of 16S RNA. However, this is just a prediction, unlike the shotgun sequencing. It's not as robust as shotgun sequencing. I do not identify fungi, virus, protozoa, and other disadvantages might be it's unable to differentiate clearly between closely related species. So what are the microbiome diversity matrices? Diversity matrices help us to differentiate between samples in the environment. So we have the alpha diversity and beta diversity. So we will discuss these ones one after the other. The alpha diversity refers to the diversity within a single sample. So this diversity tries to show us how samples in the same environment are compared, like how diverse is a sample obtained from a particular environment in particular. So while the beta's diversity shows difference between samples obtained from different environments. For example, you can compare the microbiome in the oral microbiome and the gut microbiome or fecal microbiome. So these are from different environments. So beta diversity can help tell the difference better. So the alpha diversity, there are so many alpha diversity indices, but we'll just talk about few ones that are most commonly used. We have the observed OTU. This one is simply measures how many taxas are present in the particular bacterial, in the particular sample. Like this sample has three different bacteria. Sample A has 3 different bacteria. Sample B has 2 different bacteria, for example. So sample A is more diverse than sample B. So this is how this one measures the richness. The Chow-1 index, it estimates richness, and also it's take note of rare species that are very, very rare within the within the microbiome sample. It takes note of rare species. That's the difference between the Chow-1. Although during the analysis, we usually calculate the observed OTU, Chow-1, Simpson, and Shannon index. So the Shannon index accounts for both richness and evenness. Evenness as in richness, how diverse they are. Evenness, how distributed. What's the distribution of the bacteria? While the Simpson index, Simpson, it's also all of them estimate richness. However, this Simpson focuses more on dominant species that are in the rumen. So it focuses on dominant species. These are some of the plots from a particular study showing various alpha diversity matrices, observe OTU, Chow-1, Shannon index, and the Simpson. So,
Nah, me remember HUD business. Me remember how for go fair other money on that side. Me man, them publication the way see with and then that make we get better work.
Hello.
so
It's
these
been
are
taken
the
care of. Please continue.
okay. So these are the different alpha diversity indices, like observe OTU. So this figure now shows us that in this control and treatment, these are just 2 groups. There are no difference in observe OTU, Chow-1, Shannon. So it means that the richness and the evenness are not significant, have not significantly differed from each other. So the second microbiome diversity matrix is the beta diversity. This estimates diversity between samples. It helps to explain how distinct or similar microbial communities are across different environments, treatments, or time points. So these beta diversity matrices are commonly calculated using dissimilarity matrices. So some of the similarity matrices used in calculating the beta diversity indexes are Bray-Curtis dissimilarity matrix. This compares species abundance between 2 samples. It considers how many counts are shared and how many are different. So when we are talking about counts here, we are talking about OTU counts, just like I showed you earlier. So it considers how many counts are similar and how many are different between the 2 groups. So while the Jacquard distance, this measured, it measures the presence or absence of species. Is this species present in sample A? No. Is it present? Yes. So this is what Jacquard distance measured. While the Bray-Curtis dissimilarity matrix, it compares which ones are shared between the groups and which ones are different. So the beta diversity is expressed visually via ordination technique called the principal coordinate analysis. So this principal coordinate analysis uses a dissimilarity measure. That's the one we just talked about, either Bray-Curtis or the Jacquard. Based on that calculation, it will use it to create a visual map to tell the whole story, because this calculation may not be easily understood, but when it changes it to a visualization, it will give us the whole picture of what the sample is all about. So it projects samples into lower dimensional space, 2D or 3D. This is an example of a PCOA plot from 2 groups. That is the control and the treatment group. It's very clear now that these samples within these 2, the samples within from the CON group are more diverse, while the samples from this TRT group are closer to each other. So these ones represent the replicates from the samples, while these ones are also replicates from this. So we can see how they are closely they are closely attached to each other. So differential statistical analysis. We have several differential statistical. This differential statistical analysis help us to tell statistically how 2 groups, 3 groups, 4 groups differ from each other of microbiome. So there are several statistical analysis for microbial data. However, I will talk about 2 here for the purpose of this presentation. The linear discriminant analysis effect size, also known as LEFSI. This is specifically designed to identify biomarkers that are statistically significant and have substantial effect size. It aims not only to reveal differences, but also to show how large the difference is between the groups. This LEFSI test, usually the software calculates the Kruskal-Wallis, uses Kruskal-Wallis test to identify features that differ significantly between the groups. Kruskal-Wallis is a non-parametric test, which is well suited for non-parametric data set like the microbiome data set. The Wilkinson rank sum test is also being performed. This one is to check if the changes are consistent within the replicates. So this one is optional actually, but it's being performed to check, are these changes consistent across all replicates? While the final one is the linear discriminant analysis, LDA. So all these 3 tests are performed under LEFSI. The LDA estimates the effect size of each differentially abundant feature. So if, for example, a particular taxa like the Prevotella is found to be significantly different across groups by Kruskal-Wallis test, Wilkinson rank sum test will now tell us that is this Prevotella a significant increase in this or decrease? Is it consistent across the replicates? And this LDA will now tell us, okay, what's the effect size of this increment? Is it of higher magnitude or of lower magnitude? So that's what it usually tells us. So we usually peg, for you to know the statistical difference, you usually combine 2 factors. That's the statistical test by the Kruskal-Wallis test. It usually give you p-value less than 0.05. It's usually deemed significant at less than 0.05, while the LDA is usually for microbiome data between 2 to 3. LDA score of 2, greater than 2 or equal to 2 to 3 is usually deemed significant, combined with the statistical Kruskal-Wallis test. So this is how the LEFSI test looks like. This is for 2 groups. So this one shows that the TRT group has significantly higher composition of this particular bacterial taxa. So, and from the plot, we can see that all of them are greater than 2, but less than 4. So most likely this one was pegged at 3. Most likely greater than or equal to LDA score. This is the LDA score. Greater than or equal to 3 as significant. So anything less than that, it will not count it as significant. Even if the p-value is significant, it means the effect size is not significant. So this one shows that in the con group, this particular taxa are statistically significant. So it's also most likely 3. This one is for phyla. It's also the same thing as that. That is for genera. This is for phyla. So we have another differential statistical analysis called the analysis of composition of microbiomes, ANCOM. This is a statistical method for differential abundance analysis of microbial data that accounts for compositional nature of microbiome data set. The workflow includes computation of the log ratio for each taxon's abundance to every other taxon in the data set. It computes the log ratio of each taxon's abundance. So apart from the relative abundance, it will compute the log ratio and it will compare it to that of every other taxon in the data set. That is the first one. Then it carries out Kruskal-Wallis test to check if these ratios are statistically significant between the groups. And then it now counts how many of those pairwise ratios were significant for each taxons. So these are the workflow for the ANCON. However, for the practical session, we will be looking at LEFC. Although this ANCON has been suggested by one reviewer of an article I published in one of these microbial journals. Is it one of the Springer-based microbial journals? But I have not done this analysis. But based on the on our practical session, we will be considering the LEFC. But this one is also very good. So these are visualizations that can help to give a better picture of microbiome data set. So this one is a correlation heat map between microbial genera and volatile fatty acids. This one is a relative abundance of predominant phyla in two groups. So this is to show that this particular Bacteroidota and Firmicutes are the dominant in this group. So visualizations are important in microbiome analysis to help us tell us the story in a less complicated way. So in summary, the highlights of this presentation is the strength of microbial data set lies not just in sequencing, but in interpreting it meaningfully. Without appropriate statistical analysis, we risk overstating noise and missing real biological signals. The right choice of tests depends on your data type, structure, and research question. Great visualizations help to translate complex patterns into scientific stories. On a final thought, 16x microbial data uncovers the story of miniature creatures. Statistics helps to prove the story, and great visualizations make the world believe the story. For collaborations on gut microbiome research, metagenome, metabolome, and host microbiome interaction research, animal nutrition, or related fields, I'm open to research collaborations. Let's build science together, one microbiome at a time. Thank you so much for listening.
Thank you very much, Dr. Amin. I think this is comprehensive as it regards to 16S analysis. So as we have said that it is going to be 2 sessions. So we have just come to the end of the first session. And before we go for the break, a 5 minutes break, I don't know if there are some questions or some clarification needed from our speaker here before we go for the break. If you have a question, you can put it in the chat box, or probably you can raise your hand to just ask. We'll just have 2 minutes for the questions, because during the practical session, I think everything will become more and more clearer. So I don't know if we have questions now.
Yeah, I have a question.
All right. Dr. Jimmy.
Yeah,
Please
on
be
the
very
slide.
brief.
Yeah, on the slide, there was a slide that shows the difference where you showed the treatment and control, and one group is kind of significantly higher in the treatment, and the other group seems to be significantly lower in the treatment.
Which of the analysis? Is it on the alpha diversity? Okay.
No, it's towards the end. No, move forward. Yeah, this one. Now, my question is, what if you have bacterial group or classes that are kind of similar? How is that represented on the graph? Because my understanding here is that the one on the right side is showing that the treatment has significantly higher quantity for those bacteria, while the one on the left side is showing that the treatment has significantly lower quantities for those for the red ones. So my question is, is it not possible to have a situation where there's no difference between the treatment and controls for some bacterial categories? How is that represented? Does it mean it's just going to be on the zero line?
So thank you for the question. I didn't get your name, but thank you for the question, doctor. So there is a little clarification I want to give on this figure. The red one, because it's negative, it doesn't mean that it's lower in the CON group. It means it's still higher in this particular CON group. While this green shows it's higher in this TRT group. So just to clarify that. So yes, it's possible that there will be no difference as far as left C is concerned. If there is no difference, it won't even, it will show you no difference, so there will be no plot. Is it clear?
Yeah, I think in other words, the left C analysis only shows, bacteria, species, or phyla, or any of the this taxonomy you are, running the analysis. Only show those that are significant. It doesn't show those that are not significant.
Yes.
It
Only show those that are significant.
Okay,
so you can see p-value less than 0.05.
Huh?
Yeah, based on your response now, Doc, you mentioned that it's significant on either side, but I'm wondering, what the x-axis is trying to say. What does the x-axis? What does the x-axis?
It's showing based on the LDS post.
One is negative, one is positive. So how do you differentiate between those that are on the positive side and those that are on the negative side? Since the interpretation is that both groups are actually higher in the respective treatments.
Yes. This is just for in the figure that is showing positive and negative, just to as a way of differentiating. Do you understand? However, in some left C plots, it will show you all of them on the positive side. Do you understand? So for example, now you have like in some left C plots, all of them, this one showing 4, -4, it's not -4, it's 4. It's just for illustration that it was taken to the minus side, meaning this one is significantly increased. So, but it's a positive digit, not a negative digit. This is what, unlike correlation that we will have a negative correlation, positive correlation, as far as CON group is an increase, while this particular red one is a decrease to TRT group. Proteobacter is a decrease to TRT group, right?
That's the way I'm feeling. I'm following you, sir.
Yes, it's a decrease to TRT group, but it's an increase to CON group. Don't be confused by these negative signs. They are just for illustration. In some softwares, when you plot, they will be all on the positive side. I think MicroEcho 2 plots it on the positive side. The one we are going to be considering, if I'm not mistaken.
All right, thank you.
Yes.
Okay, I think Ben has a question. He raised his hand.
Thank you very much. Can it be that, as Said was trying to ask, that the negative may give information of maybe relative to the other group, maybe is down accumulated or down, it is suppressed in terms of comparison to the other group. And then maybe the green may means maybe it is upregulated or is increased in abundance or whatever.
Yes, if what you are saying is this Fibrobacterota is an increase as far as the TRT group is concerned, while it's a decrease as far as the CON group is concerned. Is this what you mean?
Yes. In that case, if you compare the control to the treatment,
yes,
what I can understand from this figure is that
yes,
those one with the green had higher abundance in the treatment group compared to the control group.
Yes,
Whilst
exactly.
the red here also means that there was higher accumulation in the control group relative to the treatment group.
Exactly.
Okay, thank you.
Yes. So it depends on interpretation. If you say the CON, proteobacteria, if you say they are higher in the CON group, I can decide to interpret it as they are lower in the TRT group. Are we on the same page?
Yes.
Okay.
Yes.
Just to be
But
clear.
the point that what I understood from Said's question was that the projection depicts relative to the other group, not necessarily just one group, but
Yes.
what is the comparison?
Okay. Yes.
Thank
Thank
you.
you
Yeah,
for
Doc,
the
I think
yes.
one other thing that can be beneficial to all of us is that for the x-axis, we should focus on the absolute value.
Okay.
Yeah, like for the x-axis, it is the absolute value that matters and not the sign, just like you mentioned. I think that would probably be easier for everyone.
Okay. Yes.
Irrespective of
Absolute value. Not the sign. Yes.
focus
Absolute
on the
value.
absolute value.
Yes.
Yeah, thank you.
Because yes. So any more
Okay.
questions?
I think we don't even have time for more questions. So probably we will go for the break and then we just come back. Or probably if we have questions, we can take question so that we can just start by 9. We'll just start the
There's a
second
question in
session.
the chat box.
Okay.
Can you read it and
Sorry,
please drop it?
is it possible the presenter can share the last slide that has his email address?
Okay.
All this information will be sent across, please, at the end of the session. For now, let's just go ahead with the session. The information from the presenter, the PPT, the recordings will be shared.
Thank you.
Yeah. All right. Then we have come to the end of the first session and we'll go for a break. I will resume by 9 o'clock on the dot. I don't know where you are joining from, but Beijing time will just resume by 9 o'clock. So we just have a 7 minutes, break. Thank you.
Okay. Thank you, Doctor.
Dr. Amin, please,
Okay,
there's a
Prof.
question from, we're on commercial break
go.
officially. Please, there's a question from Miriam on the chat box. If you want to help her respond on the chat, that's still okay. And then
Yes.
in the
Okay.
next 6 minutes,
Can
we will
can
come
I
back.
use this?
Oh my God.
Okay, please, before we go to that, please, when you are coming back from this second session, please be ready with your R. For those that are going to follow the practical session, please be ready with your, R Studio and all the packages mentioned, in the session. So be ready with that. So that, and also, if you have some sample data, you can also, you can just come with your data, then we can just start. Thank you. All right. I can't see the question in the.
Osman, is it possible that the presenter maybe will be kind enough to share some small portion of a data set so that those of us who may want to practice alongside with him can probably move alongside with him?
Yeah, I think
I will send the data set and the R script.
Okay, thank you.
Yeah, that's great. Dr. Victoria, please, can you stop the recording of the first session because we are officially on?
All right, sir.